it's my great honor to introduce Professor Tom Rando, so from Stanford University School of Medicine. So Tom, I mean, many of you know I came from Tom's lab as a postdoctoral fellow there for 6 years. So Tom got his a bachelor degree and as well as the medical degree and his PhD all at Harvard, and afterwards he moved to Stanford and did his postdoc and subsequently become a professor there. So Tom, many of you, I mean, I mentioned it last time. So Tom was here just last year. So, and now he is our IAS senior visiting fellow. So chances that he will be here more frequently. So, but I mean, last year I mentioned this. Tom, in addition to, I mean, many of you know him as a great researcher, but he's actually a active clinician. So he spent his time in his clinics a lot. And I mean, I just to name a few things. I mean, he's the chief of service of the neurology service at the VA hospital in Palo Alto, and he is the director of the rehab center also at the VA center. He is the director of the Glenn Laboratories for Biology of Aging at Stanford University. And so I don't, I mean, the list goes on, and I won't mention too much, but to mention something that I just heard from Tom yesterday, that he just got elected to the fellow of the American Association for the Advancement of Science, so in short IAS. So he's now a elected member there. So without further ado, so please join us to welcome Tom.
Thank you very much, Tom. It's a great honor to be back, and I really enjoyed my time here last year. We had a great visit with everybody, and it was really my privilege to become a fellow of the IAS here, and my privilege to be back, and hopefully working with a lot of you in the years to come, and certainly working with Tom. So I will talk today about, so last year I talked a lot about some aspects of our stem cell work, and I thought it might be interesting today to talk about work that we do in the biology of aging. And as you'll see, there's a lot of overlap between what we study in terms of aging biology and stem cell biology, and sometimes I think we're studying one and we learn about the other and vice versa. So they're very integrally related. I'll present kind of a little bit of a historical aspect of how we became interested in this biology and the phenomenology that I'll talk about, and then I'll move toward the end to talk about the studies of epigenetics, which in a way have kind of, I would say, more dominated our more recent work, and I'll present a lot of our unpublished and some very, excuse me, preliminary studies we have on looking at epigenetic, what I call mechanisms of aging and rejuvenation, and that's obviously a very provocative word, rejuvenation, and I'll tell you how we think of that, what we mean, and I'll look forward to your feedback on some of these topics. So aging is a very curious biology, and you know, we really, from an evolutionary perspective, I would argue that we still really don't know why we age, and from a biological point of view, we don't really know how we age. And people always talk about things like the regulation of aging and the causes of aging, and I don't believe that we will ever figure any of that out as to what causes ages or what causes aging or what is the regulation of aging. But I do think we can understand the phenomenology and the fundamental aspects of how aging fits into the fundamental biology of growth and development and evolution of species. Maybe this will become clear. So what I've illustrated here is a young male or an older male, and a lot of what we study, or what are the differences between the cells and tissues of an individual of this age and this age, and clearly there's not just aging in between, there's growth and development, and then there's aging. And one of the characteristics of aged individuals, whether we're talking about humans or animals, is, and the focus of what I'll talk about, is that tissues in older individuals repair more slowly than tissues in young individuals. And if this young fellow, this is, by the way, Picasso and his son, and that's a story for another day. But if this young guy were to get an injury to his skin or a bone, he would heal very, very quickly without any scar formation and very effectively. And the older we get, the less that becomes true. And that's pretty much true across every tissue, whether it's skin or bone or muscle or heart, or basically there's a diminishing regenerative capacity of tissues in mammals, at least, with age. And that's, I would say, it's one of the hallmarks of tissue aging that characterizes this process. So, and our, excuse me, our interests in this have to do a lot with, can we do anything about it? So can we do anything about this declining function of tissues with age? And I just wanted to illustrate someone who was very successful. So this is Jeanne Calment, who is the world record holder for having been the longest on record known living human. So she lived to 122 plus years and remarkably healthy. So, we all like to think about living long, but I don't think anybody here wants to live a long time in a very unhealthy state, but we'd love to live a long time in a very healthy state. And Jeanne Calment was a very witty woman. She lived in Arles in France, and as she became older and older and became more and more famous, she was invariably visited by reporters every year, and she became 115 and 116 and 117, and she became funnier and funnier. She actually put out a CD to rap music, I think when she was 119, talking about her life. I mean, she has a really remarkable life. I mean, she took up cycling at the age of 100, and so it's just, this is what I think we would all like to think about successful aging. Okay, so we, excuse me, tend to think about aging, as I say, through the lens of stem cell biology. And I'll talk a lot about this. And I've just illustrated this slide because I think a lot about stem cells and aging in this way, in two ways. So what I call chronological aging and replicative aging. So chronological aging, if you think of a tissue in the body, let's say the brain, where the vast majority of the cells are non-mitotic. So by the time you're a teenager, you have all the neurons you're ever going to have, for the most part, in the brain. And yet those cells, over time, they age. The cells age. So that's just the experience and the changes that occur in a cell over the passage of time. And a lot of the stem cells in the body, including the cells I'll talk about, exist in a quiescent state for the most part. So they're also not dividing for a long time. And so they, too, experience this aspect of just chronological aging. They change over the passage of time in a tissue. And yet stem cells also have, what is characteristic, is they have the ability to divide and produce a lot of progeny, produce not only more stem cells, but differentiated tissue. So over the course of chronological aging, these cells are also dividing, and so they're also undergoing what we consider replicative aging. And replicative aging, in its extreme, is really what people have studied for many decades in vitro, which is the changes that occur in a cell as a cell divides over and over and over again. And then, in many cases, for somatic cells, reaching some limit where they stop dividing and they senesce. So that's kind of a paradigm of replicative aging, of this notion that the cell can only divide a certain number of times. So the interesting thing about these stem cells that are quiescent is they undergo both. So they'll undergo the experiences of the passage of time, and then they'll excuse me, undergo replicative processing as well. Excuse me, I have a tickle in my throat, so I'll cough periodically. So the tissue that we study primarily is skeletal muscle, and I'll refer to this as a single tissue, although there are hundreds of different skeletal muscles in the body, and very likely that a lot of the stem cells that we study differ between these muscles, but I will refer to this very generically, and mostly we'll be talking about muscles from the limbs and the stem cells that reside therein. So this is a cross-section of normal skeletal muscle from a mouse or a human. So each of these is, of course, just to orient you, is a single so-called muscle fiber, and these are long, elongated cells, and these are one of the true few syncytia in the human body. That is one cytoplasm and hundreds or thousands of nuclei. So these are all these myotubular structures cut in cross-section. Here's a myofiber, here's another myofiber, and you see stained in blue are the nuclei on the outside of the fibers. That's a characteristic appearance. So here's a cross-section of mouse muscle stained for laminin and white, so you see the extracellular matrix around, again, these individual muscle fibers. The nuclei are stained blue, and then this purple color stains this key transcription factor, PAX7, which in the adult is expressed virtually in every single muscle stem cell and only in muscle stem cells. So it's an extremely useful biological marker, and it's also used, the locus PAX7 is used for a lot of genetic studies because it's so specific. And if you look carefully, you'll see there is actually one of these so-called satellite cells. So muscle stem cells were originally called satellite cells because they exist kind of at the periphery. They're almost like they're orbiting these fibers at the periphery of these fibers. And in fact, if this were not stained for PAX7, it's really impossible to distinguish this nucleus of this cell, which is really all this cell is, very, very little cytoplasm, tiny little cell, mostly nucleus, from one of these myonuclei inside the fiber. So again, you can distinguish these by this staining for PAX7. Here's a higher magnification. Now laminin is stained in yellow, shown here, and here's one of these, again, PAX7 positive muscle stem cells. Now these cells are in the adult animal, in humans and mice. These are truly professionally quiescent cells. We think they can remain quiescent for, in humans, many, many years, maybe decades without dividing in an uninjured muscle. So for the most of the life, they exist in this quiescent state, again, in a very tiny cytoplasm and mostly in a nucleus. However, they have this remarkable regenerative capacity in response to an injury. And I've illustrated that here. Here's a normal muscle. Here's a couple of days after a total necrotic injury, destroying that architecture of skeletal muscle. And then these very few, they only compose maybe a couple of % of the myonuclear content, the nuclear content of muscle. These very few cells, they quote activate, which means they break quiescence, they grow, and they start to divide. And then over the course of a couple of weeks, these cells will begin to line up, they fuse to one another to form these, again, small tubular structures. Some of these are shown here. These are these small so-called myotubes. And then over a few weeks, they grow in hypertrophy and restore that normal architecture of skeletal muscle. So truly remarkable regenerative capacity, and they restore themselves. So within this field, there would be, again, one or two of these newly formed muscle stem cells. So that's their purpose in life, we think, is to maintain muscle and repair muscle in response to injury. That changes with age, as I mentioned. So we're looking at the regenerative response, an intermediate regenerative response in a young mouse, an older mouse, and a very old mouse, equivalent to a 60 or 70-year-old human and an 80 or 90-year-old human. And you see these small new myotubes being formed. There's some connective tissue in between at this point. And over the course of a few weeks, these myotubes will grow and form this normal muscle with very little connective tissue in between. But as you look older and older, you see there are fewer and smaller of these myotubes and much, much more connective tissue. So even as these heal, there'll be some restoration of the muscle architecture, but a lot of scarring remaining. And again, scar formation and impaired regeneration is a characteristic of tissue repair in very old mice or humans. So, excuse me, we became very interested years ago in why this occurs. What is it that changes with a tissue that would lead to this impaired regenerative response? And the easiest answer is, well, maybe you run out of stem cells, because of course if you run out of stem cells, you're not going to make new muscle, and that's absolutely not true. In fact, for most tissues in the body, the number of stem cells with age declines either very little or sometimes they even go up. And we quantified this, and while there is a small decline in the number over this time course, there's a much, much greater decline in function of these stem cells than there is. So you can take a young animal and deplete a lot of its stem cells that will still regenerate perfectly well. So it's not a matter of number, it's definitely a matter of function. And this is a figure from work that Irina Conboy did when she was a postdoc in the lab many years ago. We had been studying the biology of these stem cells, and we knew that it was essential that the Notch signaling pathway is induced to turn on in these cells to induce a normal regenerative response. We also knew, data I won't show you, that with age, one of the failures of the regenerative process is there's a failure of activation of this Notch pathway in these aged muscle stem cells. So Irina had a very simple-minded experiment, which is, well, what if we induce Notch signaling artificially in these aged stem cells? What effect would it have? So these are two old mice, or muscles from old mice, and this is the control where she produced a focal injury here. This is uninjured muscle. So you see this regenerating, poorly regenerating old muscle. But at the same time she induced an injury, she injected an activator of the Notch pathway. She could get this muscle to regenerate basically just as well as a young muscle. Excuse me. So that really changed our perspective on this, because this really said to us in a very simple experiment that this age-related change is not permanent and irreversible. This may be an experimental intervention, but at least from this very simple approach, one can make an old muscle regenerate using the stem cells that are already there as well as a young muscle. So it basically said that this phenotype of this impaired regeneration was in some sense reversible. And so it really started us down this thinking of, we're not looking to replace the stem cells, we're just looking to change the signals that they receive. And if you did that, could you in fact then make old tissues repair and regenerate as well as young tissues? So we went from this very focal approach of just injecting a particular activator in one tissue to induce the normal activation of old stem cells to a much more systemic approach using this technique called parabiosis. And this is a technique where two animals can be joined to one another surgically such that they develop a single shared circulatory system. And this technique has been around for over 150 years, and many important discoveries in endocrinology and immunology have come from this technique. So we reasoned that maybe the reason why old stem cells don't activate in response to injury is because they're not exposed to the same youthful circulating environment that young stem cells are. So we created these parabiotic pairs, either so-called isochronic, same age, young and young, or old and old, or the real experimental mice, which are these heterochronic pairs, so a young animal compared to an old animal. So as I mentioned, the goal here is we surgically connect them, and over the course of a couple of weeks, they develop a single shared circulatory system. So after, say, 6 weeks, this animal has now been exposed to the circulating environment of the young animal, and the young animal has been exposed to the circulating environment of the old animal. And the question is does that parabiotic influence, does that change the biology of tissues in the old animal and or the young animal? And so what we did is we did this in a series of different tissues, but in one case we would, for example, keep them in these pairs, and then after 6 weeks, injure the muscle of the old animal or the young animal and ask how well did they repair. Excuse me. So in these isochronic pairs, the young muscle is regenerating quite well. Here's uninjured muscle, here's injured muscle, just like an unpaired young animal would. In the old isochronic pairs, so old to old, here's the uninjured tissue, here's the injured tissue. Again, you see a little bit of regenerative myogenesis along the border, but a lot of scar tissue formation. But if we looked at the reparative process in the old parabiont in a heterochronic pair, so an old animal that had been exposed to the circulating environment of a young animal, we saw that the muscle regenerated just as well as a young animal. So this was really, again, this kind of really extended this idea that if given the right environment, the right signal, can an old tissue be essentially induced to regenerate just like a young animal? And this was the case. We saw this, we looked in muscle, we looked in liver, we looked in brain, and then very briefly in a few other tissues. And in every case, we saw the same effect, which is the old tissue, in terms of regeneration assays or even homeostatic turnover, now looked much more like a young animal. Now, there was no free lunch here, so if you look at the young animals, they all regenerate less well than the unpaired young animals. So there's clearly some crosstalk going on here between the circulating environment of the young animal and the circulating environment of the old animal that's modifying the behavior of the tissues in the other parabiont. So if we looked at the active markers of Notch signaling, here we're looking at expression of the Notch ligand delta. Here's the young animal, the cells from the young animal and the isochronic old animals. Notch signaling is much higher in the young versus the old. If we look from these heterochronic pairs, it's intermediate in each case. So Notch signaling is induced in the old animal and actually suppressed in the old animal. A few years later, in collaboration with Tony Weiss-Corey, our colleague at Stanford, we went back and looked more carefully at the brain, and we saw basically the same phenomenon. So here we're looking at neurogenesis, shown by BRDU uptake, labeled here. And these are the young animals. You see a lot of BRDU incorporation. In the old animal, there's a marked decline in neurogenesis with age, which has been well known. But if we look at the heterochronic pairs, there's a substantial induction of neurogenesis, really as effective as any intervention that has been shown in terms of inducing neurogenesis in the brain of an old animal. So the same effect in terms of this heterochronic pairing. So this approach has been used now many, many times by other investigators to look at the effects of heterochronic parabiosis on the biology of aged tissues, either in normal conditions, homeostatic conditions, or in response to regeneration. And a lot of tissues have shown the same thing. So muscle and liver, brain, this is a spinal cord regeneration, pancreatic beta cell regeneration, actually just heart size, brain vasculature, and more recently, bone healing. So it's been proven to be a very robust phenomenon across many tissues. This leads, really led us to this question, and what I'll talk about for rejuvenation. Is aging reversible? Meaning, if you have a phenotype in an old tissue that has changed along some axis from a young tissue, is it possible to actually reverse that process, not just change the behavior of the old cells or the old tissues, but actually move them back along the same axis they moved along in going from young to old? Is there really a reversibility of this process? This is just a painting from the 17th century Weimar Republic, which is a picture of the Fountain of Youth. So the older individuals, they're all women for some reason, are being carted in, by these carts. They enter the Fountain of Youth and they emerge as young women, enjoy, love and food, great food, and all the great things of youth. But really the question is there anything that really can not just enhance function, but actually reverse the phenotype of aging cells and tissues? So I want to just give you a couple of other examples besides this, this very extreme paradigm of heterochronic parabiosis that I think also illustrate this notion of reversal of an aging phenotype. And I want to illustrate this in particular because in the aging field, in the biology of aging field, there are a lot of interventions that are done regularly that are done to test the ability of a drug or a treatment to slow or delay aging. So the classic is caloric restriction. So if you put animals on a calorically restricted diet, they will live longer, they will maintain kind of healthful living longer, and that appears to, quote, slow aging. But that's an intervention that's done throughout a lifespan that has an effect that appears to be slowing or delaying aging. A lot of drugs that are being tested, same thing, administered chronically to animals that appear to delay the onset of aging phenotypes. But what we're talking about here, the difference is we're taking animals that are already old, that already manifest aging phenotypes, treating them, and I'll show you a couple of other examples, such that the cells and tissues now appear younger. So that's very, very different than an intervention that would slow or delay aging. So this is work done by Howard Chang, a colleague at Stanford, now quite a few years ago, in which he had shown that with aging in skin, there's a marked induction of a signature of NF-kappa B signaling, as if with age there's an increase in NF-kappa B signaling in the skin. And what he wanted to know was whether this was actually driving an aging phenotype. So he created a mouse in which he could induce the expression of an NF-kappa B inhibitor late in life. And this is not delaying. So he, excuse me, he created a mouse in which he could induce this I-kappa B, an inhibitor of NF-kappa B, in already aged skin. And so here he has, these are young and old, and these are the control animals, and these are the animals in which he's given tamoxifen to induce the expression of this NF-kappa B inhibitor. And he's looking here at, excuse me, at this marker of senescent cells, beta-gal shown in blue, and he shows that when he turns on this inhibitor, it's markedly diminished. There's an increase in the proliferation and actually an increase in skin thickness. So again, this is taking an already old animal and making the old skin now appear biochemically and anatomically like younger skin. This is work from the Zheng lab at the University of Michigan, in which, again, looking now at hematopoietic stem cells, which are known to have very clear age-related phenotypes, and then administering to old animals this drug rapamycin. And for those of you who read any of the aging literature, biology of aging literature, rapamycin is one of these drugs that has been shown to have remarkable effects in extending lifespan in a whole variety of organisms. And can, again, delay the onset of these age-related phenotypes. But in this case, they administered rapamycin to already old mice. Okay, and then they isolated the hematopoietic stem cells from either the control or rapamycin treated, and then in these competitive reconstitution assays asked, what is the phenotype of these already old hematopoietic stem cells that have been treated with rapamycin? And here you're showing the control, the young, the old, and those old now treated with rapamycin. In almost all of these assays, the cells that have been treated with rapamycin now exhibit a more youthful phenotype. So again, not a delaying of an aging phenotype, but actually taking an old cell and inducing it to become, apparently, in many ways, like a younger cell. So my colleague Antonio Weiss-Corey has taken this beyond parabiosis and asked, well, if we can assume that the effects of parabiosis is through the circulation, what would happen if you just take blood from a young animal and administer it to an old animal? And this is done as a series of injections over a course of several weeks. So either blood from a young animal to an old animal or blood from an old animal into an old animal. And again, looking at the brain, looking at aspects of neurogenesis, as well as functional tests, cognitive tests in these old animals, they were able to show remarkably that the administration of young plasma to old animals showed increases in neurogenesis in the old animal compared to old plasma, as well as increases in cognitive performance. And this is just a water maze test in which what they're measuring here is the number of errors as the mice learn. So that as they learn, they get better, fewer errors. And it turns out that the mice that were given the young plasma, shown in red, going from one day of training to the next day of testing, remember the task better and continue to learn, whereas the animals that got the old plasma basically behave like control animals. They quote, forget and have to relearn the task again. So they've looked at a series of behavioral tests and shown that this administration of young plasma really can have a comparable effect to what we see with heterochronic parabiosis. So this leads us to kind of the hypothesis that we've now been testing for several years about this idea of reversibility. And that is that cellular age or tissue age is actually, to some extent, an epigenetic state that is modifiable and potentially reversible. So that's a profound hypothesis that if it's right, it's going to be important. So a corollary of that is that these rejuvenated cells, that is cells in these heterochronic parabionts or treated with rapamycin or in which NF-kappa B is inhibited, are actually equivalent or moving toward really the phenotype in every way of a young cell, and that this rejuvenation occurs, this is again a corollary of this hypothesis, by cellular reprogramming, or what we call epigenetic rejuvenation. So there's something that's actually modifiable in an epigenetic way in an aged cell to make it more like a young cell. And I just want to really focus on this point of what do we mean by cellular reprogramming and epigenetic rejuvenation. And to illustrate how we think about this, I want to take a page from the book of induced pluripotent stem cell biology. So familiar to everyone here is you take a very differentiated cell, and through the administration of a set of genes or small molecules or different ways, one can induce these highly undifferentiated, highly pluripotent cells, these iPS cells, which then themselves can be redifferentiated by inclusion in a blastocyst to make a mouse. So these cells, and you can do this over and over again, so they can acquire this differentiation phenotype, become undifferentiated, and then redifferentiate again. And that's a well-known, well-established paradigm. Well, I would argue that there's something else equally important going on here that receives much less attention, and that is if you take these cells all have some kind of age to them, however you want to define or quantify cellular age, and then in response to this induction of pluripotency, they become young again. Because by whatever criteria you want to define cellular age, these cells are as young as they get. They can make an entire new animal, and you can do this over and over again. So these cells become, old and then young and then old again. And this is all coupled to this process of dedifferentiation and redifferentiation. And in fact, that's what really the fundamental biology behind somatic cell nuclear transfer depends upon. This is Dolly, the first cloned mammal, a sheep. And again, you take a nucleus from a cell of an adult, and that, again, cell has some age. You implant it into an oocyte, and now you've created a cell in which that aging clock has been reset back to zero. And you can make an animal, and that animal can reproduce. And so without that, you would not get otherwise a normal sheep. Now, people always ask, well, Dolly wasn't completely normal, and it's true, there were some autoimmune aspects, but there was no aspect that we know of that Dolly was older than a sheep of her chronological age. And I would argue that actually this notion of epigenetic rejuvenation is so deeply embedded in our understanding of biology that we just don't even think about it in the most fundamental way, and that is, so this is a fertilized egg. Okay, so if you think about the process of fertilization, say in humans, you take two cells, sperm and an egg, which in humans would at least be decades old, however you want to quantify them. You put them together, and you produce a cell that by definition is starting over again at zero. If not, every time there was a fertilized egg, you would inherit some aging from your parents, and the species would quickly pass to extinction as children acquire aging phenotypes. Well, they don't. With every fertilization, whatever that aging clock is reset to 0. Okay, so clearly this happens. It's not whether, so this is not a hypothesis. Clearly this happens and this has to happen. And the question is, in this case of, where I'm using these examples like iPS cell, somatic cell nuclear transfer or fertilization, this rejuvenation is always coupled with dedifferentiation. And the question is in these experimental paradigms, are we actually rejuvenating cells without dedifferentiating them? Meaning, if we do this heterochronic parabiosis, we're not making muscle stem cells into pluripotent stem cells, we're just making them into younger muscle stem cells. The skin is not becoming pluripotent, it's just becoming younger. So is it possible to uncouple these two processes, and is that what we're doing in these experimental interventions, what looks like rejuvenation? Hope I can make it through, sorry. So we've really focused on a few different things. I'll talk about 2 of them. I'll talk briefly about this, which is in this parabiotic paradigm. I just want to talk about this briefly. What are these systemic factors, or in this blood transfer, what are the systemic factors that mediate these changes of stem cell functionality during aging and rejuvenation? Can we begin to deconstruct the components of blood or plasma that are actually influencing cells that are creating this kind of apparent rejuvenation? And what I'll spend more of the time talking about is, well, what changes inside these cells that occurs in these cells with aging that can we understand about the biology of this process of rejuvenation? Can we begin to understand this at a molecular level? Okay, so first of all, we've looked a little bit at this idea of looking for factors in blood that change with age, that are altered with parabiosis, and that affect cellular function. And so this is again with Tony Weiss-Corey's lab. We looked at a series of proteins that increase from young to old, that change with parabiosis, and that are restored more to a youthful level in the old parabiont circulation, and came up with this group of cytokines and related molecules that increase with age. And so we've tested a bunch of these in these models of stem cell aging, and this is just one example from that paper we published in 2011, which we took this one molecule, CCL11, which increases with age, and actually if you administer CCL11 and we're looking here at neurogenesis using non-invasive imaging. So this is in the control. You see no difference over the course of 4 days. But in the animals that receive CCL11, you see a decrease in this neurogenesis activity, and that can be measured in tissue sections as well. So clearly this is a protein that can mimic the aspects of aging, and if we neutralize some of these, we can restore in old tissues and old brains a more youthful neurogenic activity. So that's the kind of paradigm we're looking at, is factors that are present in blood that change with age and whose either gain of function or loss of function can mimic this aspect of aging and rejuvenation. So we and others have looked at a lot of now factors. I've listed a few here. Some of those that appear to be aging or deleterious factors like Wnt, TGF-beta, CCL11, beta-2 microglobulin, and then so-called rejuvenating or beneficial factors. This myostatin like TGF-beta like protein GDF11 was reported several years ago. This remains a little bit controversial at this point. And then oxytocin reported again by Irina Convoy in 2014. So one can begin to pull out factors that change with age and that influence the biology of these cells. And the question is, to what extent are these going to be some cocktail of factors, and which ones are going to be the positive effects and which ones are going to be the negative effects? Okay, so I don't want to talk about that anymore. I'd like to switch to, so what changes in a cell? What makes a cell that is otherwise behaving and molecular like an old cell become more like a young cell? And this is what really led us to focus a lot on epigenetics. And really, it comes down to a couple of important questions. So I said as a corollary of this hypothesis is that a rejuvenated cell would have to then be like a young cell. And the question is, how similar are they? So if we induce this rejuvenation process by whatever mechanism, can we then say, how has that cell become young? And really it comes down to this fundamental question is, how do you define the age of a cell at a molecular level? Can you get a readout, whatever the readout is at a molecular level, and say, if I get this phenotype, if I get, this kind of pattern, that is a young cell versus an old cell. So that's something we've been striving for. I don't think we have it, but really to be able to say, we can define cellular age at a molecular level. And I think if we can achieve that to some extent, it will allow us to test these hypotheses much more rigorously. So we've looked at a couple things. I'll talk a little bit about work we've done looking at transcriptional states, and then I'll finish by talking about our ongoing work looking at chromatin and epigenetics. So this initial work is done primarily by a senior fellow in lab, Ling Lu, who's really spearheaded this work. And so she started to look at this aspect of transcription. So what we're looking at here is transcriptional profiles of either young cells on this axis or old cells on this axis, just from young and old animals. And these are these quiescent muscle stem cells, freshly isolated by flow cytometry, and then just looked at their levels of expression. And what you see is that remarkably, young and old cells are very, very similar in themselves. So really along this diagonal means they're identical, but very, very similar. It's not as if young and old cells are extremely different transcriptionally. But interestingly, in these heterochronic pairs, the young and old cells from these parents are even more similar. Okay, so there's much less scatter if you look at the comparisons of levels of expression from these heterochronic pair bonds than if you just take the comparison for a young or old animal. This is also shown here. So it's just a heat map from some old microarray studies in which you look at old cells and young cells. Again, they're really very similar. There are some interesting differences, and we've focused on some of these, but they're very similar. And yet the profiles from the either young or old heterochronic pair partners are more similar to each other than they are to either the young or old cells. So again, suggesting these cells are becoming more similar as opposed to in any way different from each other. So another way to look at this is to just ask, okay, for all of the genes that we know are downregulated with age, so shown in this column, if we look at those genes now in the heterochronic pair bonds, how many of these in the old animal actually are restored to a more youthful level? And almost all of them. If we look at genes that are upregulated with age, what percentage of those are decreased with this heterochronic parabiosis? And about 80% of them. So again, it's suggesting the phenotype of these cells is becoming more intermediate between young and old than moving it away from the state in which they were in before this intervention. So this is principal component analysis. Again, looking at these populations of these stem cells, and here you see a group of young cells shown here and a group of old cells shown here. So clustering very tightly with each other, but still distinguishable by PCA analysis. And so we consider this, in a sense, the aging axis, because these cells are going from young to old. And if we look at the profiles from the heterochronic pair bond pairs, here's the young and here's the old. So they're displaced off this axis by the parabiosis. And we know this happens even in the isochronic pairs. They're displaced off this axis. And yet, they're sort of intermediate between the young and the old along this aging axis. So all of these keep telling us the same thing, is that in this paradigm, these cells are moving toward each other actually along an axis, along an aging axis, as if the young cells are becoming older and the old cells are becoming younger. Okay, so that's what we've been finding with this transcriptional analysis. But then to define more clearly what it might mean for a cell to be young or old, we've really turned to looking more at the state of the chromatin. And the first question we had is, if we're right that there's kind of an aging axis and cells go from young to old, is there a histone code that defines cellular age? And if so, is it something that we might be able to extend not only to this stem cell population, but to other stem cell populations and even to non-stem cell populations? So we started doing this by just looking at modifications of histones using ChIP-seq analysis in these young and old muscle stem cells. So as you probably know, there are innumerable modifications that have been found on histones across these proteins, methylations, acetylations, phosphorylations, ubiquitinations. So one could clearly spend a lifetime studying these modifications. We focused on a few of the most well-studied histone marks, and I'll talk about two in particular today, and that's H3K4 trimethylation, H3K27 trimethylation. I'll come back to that. So here what we're looking at first, so we're just comparing these young stem cells to the old stem cells to ask, from a point of view of histone modifications, can we find a difference between these populations? So this H3K4 trimethylation, as probably many of you know, is a modification that occurs when it occurs at promoter regions of genes, at transcription start sites, mark genes that are either actively transcribed or poised to be transcribed. So a marker of genes that are either active or activatable. Whereas this H3K27 trimethylation at transcription start sites tend to occur on genes that are repressed, so gene repression, so not expressed. So we have one active mark and one repressive mark, and when we look again across the whole genome, all the genes at transcription start sites, we see that in the young cells shown here in yellow, there's a high enrichment of this active mark across the genome in a lot of these transcription start sites. Also true in old cells, diminished to some extent, but still a lot of enrichment of H3K4 trimethylation across the genome. What was surprising is when we looked at this repressive mark, H3K27 trimethylation, barely present at transcription start sites in the young cells, but in the old cells, markedly enriched. So a huge increase in this repressive mark across the genome at transcription start sites. Now what's interesting is that for the most part, and I'll come back to this, most of the genes that show an enrichment going from young to old of this repressive mark are genes that are already not expressed in the young cell. So this is not, for the most part, associated with a change in gene expression, and yet they're acquiring this repressive transcriptional or epigenetic mark. So we're not sure what this means, but I'll come back to this in a minute. This is just another way of looking at it. Again, across the genome, all the genes are shown here again horizontally. Here's a transcription start site. Looking at this activating mark in young cells and old cells, they look very similar by this kind of analysis. But if we look at this repressive mark, very little evidence of enrichment in the young cells, marked enrichment in the old cells. So clearly, at least among these two, this is the dramatic difference between young and old cells, is this acquisition of this repressive histone mark. So we were working on at the same time, from a purely stem cell biology point of view, we were studying the epigenetic changes that occur when these quiescent stem cells activate and begin proliferating as progenitors, just as we understand the process of how these cells regenerate muscle and what they do. We were also looking at this process of going from quiescence to activation. And when we look at this same mark, H3K27, we found the same surprising enrichment in the quiescent cells shown here. These are from young cells, very little enrichment, as I already showed. But as these cells activate and begin dividing, marked increase in this repressive mark across the genome. And just like in going from young to old, this enrichment occurs, for the most part, in genes that are already not expressed. So there's something about aging as well as replication that seems to induce this acquisition of this repressive histone mark across the genome that's, for the most part, not associated with a reduction in transcription. So we're not sure what it means, but it turns out to be a very reliable marker of both cellular aging and cellular activation from this quiescent state. Again, this is just another way to look at it. This is comparing young versus old quiescent cells using this repressive mark. As I said before, very little similarity. But if we look at old quiescent cells, these are the cells that have become old, compared to young proliferating cells, they're remarkably similar. As if somehow with age, there's something about now cells in the quiescent state that resemble the behavior of young cells that are actually proliferating. So we're not sure what this means, but it's really taken us this idea of some similarity back to this image of, is there something about the aging of the stem cell population that is actually not only undergoing aging, but is actually integrating maybe the replicative history of these cells. And so we're testing this now by whether if we induce these cells to undergo multiple cell divisions, do they seem to age faster, essentially? Do they seem to, because they're dividing and returning to quiescence, is this somehow a cumulative history of the replicative history of these cells? We don't know this yet, but that's what we're testing in terms of this similarity between old quiescent cells and young proliferating cells. Okay, so let me just turn and end with some more work that we're doing now on this idea of the epigenetic state of cellular aging. And I mentioned that this acquisition of this repressive mark, for the most part across the genome, occurs in genes that are not expressed. But there were clear examples where the acquisition of this repressive mark was clearly associated with a reduction in gene transcription. And notably, that occurred actually at the histone genes themselves. So here's a cluster of histone genes, which actually have this active mark in the young and the old, have no repressive mark in the young, but with age, they acquire this HEK27 trimethylation at the transcription start sites in all of these histone genes. And in fact, when we look at the transcriptional level, all of these histone genes, levels of transcript are decreased. So here's a case where the repressive mark correlates with a reduction in gene transcription. When we look at the protein level, in fact, looking at H3 and H4, there's a reduction in protein levels in histones. So this was very interesting to us because, at least in yeast, an increase in histone gene expression is associated with an enhancement of replicative lifespan, and a reduction in histone gene expression is associated with a reduction in replicative lifespan. So we were wondering in this stem cell population if changes in histone levels might also be an important biological feature of cellular aging. So we've gone on to look at this and asking, what does it mean actually that you get a reduction in histone levels in the cell? What does that mean in terms of the biology of these cells and the state of chromatin if you're actually losing histone levels over time with age? So we've looked at this using ATAC-seq to look at chromatin accessibility in, again, young versus old cells. And this is just a browser view looking from this region of chromosome 10. And you can see actually, looking at chromatin accessibility, you see these peaks very similar at transcription start sites in young and old cells as predicted. But if we compare them globally, we see again that there's a clear segregation of young cells versus old cells by ATAC-seq analysis. And if we look at, in more detail, at nucleosome positioning, so you can clearly see, at least across this length, kind of clear peaks of a single nucleosome, a second and a third. If you look at this region here, this is the nucleosome-free area of the chromatin, and there's much more area of nucleosome-free chromatin in aged muscle stem cells. So we're hypothesizing and looking at whether actually the reduction in histone levels is actually leading to an increase in nucleosome-free levels, and whether that actually biologically is influencing the behavior of these cells as they age and their decreased ability to activate in response to stimuli. So finally, I just want to mention a couple of other studies we've been doing. Obviously, there's a lot of interest in the relationship between DNA methylation and chromatin accessibility, and in particularly nucleosome occupancy at genes. And so we've just started to look at DNA methylation patterns in these young versus old stem cells to see if there's also a clue from these methylation patterns as to what changes with age in these muscle stem cells. So this is all work done by Jamie Brett, a graduate student in the lab. Fanciful picture of her here. And she's done whole genome bisulfite sequencing on these cells and has just begun. I'll just show a couple of preliminary data she has on looking at the methylation patterns in these cells. So again, like transcriptional analysis, like histone modifications, like ATAC-seq, you can clearly segregate the stem cells by age according to their DNA methylation patterns. So there's a clear change over the lifespan of DNA methylation patterns. And Jamie's gone to kind of analyze this in detail. And interestingly, what she finds in muscle stem cells, which is contrary to what people find in a lot of whole tissues across age, is that by and large, there's a hypermethylation of the DNA in muscle stem cells with age, really across a whole series of different regions of the genome, whether it's CPG islands, repetitive elements, gene bodies. But a couple of very interesting areas are profoundly hypomethylated. So the question now that we have is, what is regulating the relative hypomethylation, and to what extent does any of these methylation patterns correlate with what we see in terms of chromatin accessibility by ATAC-seq? So I just want to end there and just again return to this question of, if we're looking at these epigenetic changes and we're trying to study the epigenetic regulators that might modify them, so whether they're regulators of chromatin modification, regulators of DNA methylation, methyltransferases, demethylases, can we begin to understand not only the molecular phenotype of these cells as they go from young to old and perhaps rejuvenated back to young, but how do we begin to understand which of those regulators are modifying the chromatin in these cells that's leading to this rejuvenated state, and can we begin to really answer this question about the reversibility of aging and the molecular phenotype of young cells, old cells, and these interventions that appear to reverse this aging phenotype. Okay, so I just want to end by acknowledging people who did this work. So I mentioned, obviously, Ling had done a lot of the work on the early transcriptional work and epigenetic profiling. Jamie is working on this, the DNA methylation patterns. I should have mentioned Peter Bielen is a bioinformatician in the lab who's been helping with the analysis of all of these data sets. Tom was very involved, I should have mentioned this, really early on in really all of our studies of these quiescent muscle stem cells, both in terms of developing the techniques for purifying them, which really set us going, and then also really leading us more into the world of bioinformatics that we hadn't done before. I mentioned Irina Conboy, who pioneered the work on parabiosis. Tony Weiss-Corey is our close collaborator. We work on most of the neurogenesis and the neuroscience aspects of aging and rejuvenation. Anne Brunet is a colleague with whom we do a lot of work on just stem cell aging and epigenetics. And Peggy Goodell is our close collaborator at Baylor, with whom we work. I didn't talk about any of this work today, but looking at hematopoietic stem cell aging as well. And I'd just like to, of course, acknowledge the funding sources that have really supported a lot of this work over the years. So with that, I'll stop and I'm happy to take any questions. Thank you.
I have a question about during the rejuvenation process, some external factors from the young cells are introduced to the old cells and makes it and turns back its biological clock. So my question is, after stopping this rejuvenation process, stopping administration, those stuff, will the rejuvenated cells age? The aging process, will that be faster than the native young cells at similar age?
Would it be faster than? So it's a great question. So we've done the experiment of uncoupling the parabiotic mice. Okay, now we haven't done a lot of these, and of course, it's, these assays are not highly quantitative. But I would say this is that when we uncouple the mice, the phenotype of the tissues returns back either to the young state or the old state over the course of a couple of months. So I think there are two important things. Number one, they return back to the original state. So the effect that is induced by this parabiosis is not permanent. And I suspect that's also true of the other interventions, the NF-kappa B blockade and the rapamycin treatment, but it hasn't been done. So I think these interventions are required to maintain this. So these are states that are reversible, but they're not stable. They're not stable in the absence, at least as far as we know, of continued intervention. So that's the key point. I don't know if they age faster, but they certainly return back to the original state. But the other thing that's interesting is they don't return immediately back. So we've done, again, just a couple of time points over the course of a couple of months of uncoupling the mice from the parabiosis. And after a couple of weeks, you still see the effects of the parabiosis. So there's some molecular memory there. There's something that takes a while to forget, several weeks, if not several months, to return back to the baseline, but they do return back. That's a good point.
Thank you so much for your talk. I have a question because you mentioned that there is some signaling pathway like Notch, Wnt, NF-kappa B that was declining. So this will lead to transcriptional regulation. But you are more interested in epigenetics. So I just wonder which is the cause, which is the result.
So that's a very good. I mean, I don't know the answer to that. I mean, so the reason why we focused on epigenetics is partly because of this idea of the memory of the kind of the molecular memory that is, it's somewhat perdurant effect. I mean, not that transcription factor networks can't also achieve a stable state, but it's really this idea of kind of a reprogramming of the cellular state. So I don't know whether the initial link between the intervention and the cellular phenotype is a transcriptional one that leads to an epigenetic change, or if it's an epigenetic one that leads to a transcriptional state. So we don't have that worked out in any way, and it could be either one. And I think ultimately we would need to kind of block. We would need kind of loss of function studies to either block the chromatin regulators and see if that's downstream of a transcriptional activator, or block the transcriptional activator and see if that's downstream stream of chromatin modifications. So we don't know, but that's, we're very interested in that question. It's a good question.
Thanks for the nice talk. So I have a question for your parabiosis study. So if you conjugate the young and the old mice, so without any injury, do the old mice get better muscles? For example, the central nucleate fibers or fibrotic tissues.
Yeah. So it's a tough question. So the answer is I don't know. I mean, but it's a tough question to answer because there's not that much wrong with the muscle of a 20-month-old mouse. That is, it's not very fibrotic. There are very few central nuclei. So if you don't, so part of the reason why we do the injury is because it probes the stem cell function very, very dramatically, right? That if you don't do that, there's not much to measure. So it becomes a real tough assay of sensitivity in a tissue that's quite stable and doesn't differ too much between a young and old. So you'd have to look at a tissue in which either the anatomical or the molecular phenotype between a young and old animal was really quite very, very different and ask that question. So I would just point to two other tissues, though. So we looked at brain and we looked at liver without any injury. Okay, so the brain, the decline is in spontaneous neurogenesis. That's not an injury response. That's just spontaneous. And that's improved with parabiosis. In liver, we're looking again at turnover of hepatocytes, spontaneous, not without injury. And that improves from the old in the heterochronic parabiosis. Muscle is just they're just not a clear phenotype. They're not very at all atrophic at that point. So it's just a matter of sensitivity to know what to measure, to ask if there is a sort of restoration of more youthful function in the basal state.
But the reason why I ask this question is that during the injury, it's not only the blood from the young animal that come to the injury site, but also the immune system, the immune cells, macrophages, other things that came from the young mice that maybe also contribute to better phenotype.
Yeah, this is a good question. So it's certainly possible. And what I didn't mention is when we did all these early studies, we always used young animals that had a constitutive GFP transgene. And what we know is that the regeneration of the old tissue was done by resident cells in the old tissue. But that doesn't mean what you're suggesting is not true, that circulating cells from the young animal could pass through the tissue through secretory factors, affect the old tissue by the young immune cells. So that was always possible, but and that may still be true. But what's also true is that if you just take young plasma, okay, so that's you've removed all the cells, you can reproduce at least some of these effects by plasma transfection. So that says it's not, it doesn't require the cells, the immune cells, infiltrating cells of the young animal to see the effect. They may also contribute, but it's achievable by plasma alone.
You said you see increased H3K27 trimethylation in old muscle satellite cells. Have this been seen in other aged tissues?
So we've looked a little bit, and again, our colleague Peggy Goodell has looked at this and does not, well, she sees this to a much less degree in the hematopoietic stem cell aging. But not to the degree that we've seen. And in fact, when she's done whole genome bisulfite sequencing and looked at DNA methylation, again, the results are there are some similarities, but they're different. So we're actually trying to figure out, like right now, if we compare data sets and we're going to reproduce all of these and do them simultaneously with the same assays in the same person, and number one, make sure it's not just something technical, and if there are signatures in both histone modification, DNA methylation that are shared, that are important. Right now, I would say they're quite different, actually, surprisingly, and we have to figure out if there is something underneath these that is shared. And we're looking at a few other stem cell populations as well. But it's not as if this is the signature by any means. Yeah.
Another question is, have you looked at the H3K9 trimethylation? In other words, whether there's changes in the heterochromatin state in the aged satellite cells?
Yeah. So we have looked at that, not in as much detail. And there is an increase in H3K9 di and trimethylation enrichment in old versus young. And you do see a greater number of heterochromatic foci, by staining, so there by other measures as well. So it looks as if there's more heterochromatin in the old cells, but not nearly the difference that we see with HCK27 trimethylation.
I'll ask a question also. So for the attack-seq analysis, so you indicated that the old satellite cells has more nucleosome-free regions in the. So these regions, have you looked into the whether or not there are some, say for example, regulatory region? In other words, are there some kind of gene expression that all of a sudden get de-repressed?
Right. So we're just doing the analysis now, and so we've looked. First of all, we looked at at transcription start sites, which are typically nucleosome free, and and asked whether that's a big, and that's not the primary component of the increase in nucleosome, but I don't have an answer yet as to what the characteristics of that nucleosome-free region that characterizes the old cells. And so I don't have any also correlation between the genes we know that are changed and that yet. So I don't know yet, but that's exactly what we're looking for.
What about the DNA methylation, like the hypo
correlation.
or the hyper?
So again, we haven't gone back to, that's what we're just starting to do that now, because we had these DNA methylation data for a long time, and we just got the attack-seq data a couple of weeks ago. So we're just going back to look at this now. Yeah.
Well, thank you for your talk. I just have a simple question. Do you think that the aging of central cell is a cell autonomous process, or it's a process that's induced by extrinsic factors? Because according to your parabiosis experiment, it seems that there are factors that circulate by other cells that control the aging process of central cell.
Yeah, I guess I would answer that question by saying that almost certainly both. I know that's sort of waffling, but here, so there's, for example, data that if you take these old cells and you transplant them into young hosts, they will retain at least some aspects of their aged phenotype. So suggesting a cell autonomous change that occurs with age. Now, and when we did the parabiotic work, we never claimed to be even asking this question about whether it's either or. The point of the parabiotic experiments, I think, is that the extrinsic environment can certainly influence the behavior of the cells, whether or not there are cell intrinsic changes. And the thing that I think is just very difficult to ever tease out is, if you find, quote, cell intrinsic changes, and what does that mean? Usually that means that you put the cells in vitro and they kind of retain some characteristic, or you transplant them and they retain some characteristic. And so you call that cell intrinsic or cell autonomous. But how do you ever say that change was not induced by the environment in which they were in for a long time? So I try not to get too much into whether it's either or. I would say that you can see what look like cell autonomous changes that are sustained, at least for a period of a few days in vitro or a few days in vivo. And you can say that the cells, the features of the cells are modifiable by the extrinsic environment. So I guess I really try not to say it's either or. I really try and think, what are the cell intrinsic changes and what drives them, and to what extent are those changes driven by extrinsic influences, and at least some of them are.
Thank you.
Thank you for your talk, and I missed a few points, and I'd like to ask what is the tissue responsible for secreting this juvenilizing factor, and is there any tissues that age faster than other tissues in your study?
So sorry, your first question, what tissue secretes which factor?
The juvenilizing factor that's responsible for reverting the age.
Oh, yeah, so, I mean, I wish we knew. So again, we've looked for factors, right? We've looked for factors, and for a variety of reasons, we've actually looked more for aging factors than for rejuvenating factors. It's not, even the people who have looked for sort of more rejuvenating factors, it's not clear whether it's coming from the liver or from blood cells. They tend to be expressed in a lot of tissues. So we are exploring this right now to try and figure out, just in general, when we look at the proteome of blood, where the factors are coming from to try and get at your question. But I don't really have a good answer for either for the aging factors, which probably come from inflammatory cells, or for the more rejuvenating factors. Your second question was, what was it again? Sorry. Oh, yeah. I mean, again, that's a funny. That's that's an interesting question, and I guess it just depends on what you consider aging faster. Because I mean, the way I think of it, and this is not a very popular way to answer this question, because I think the popular way to answer the question is to say, well, there are tissues do age at different rates, and pointing to glomerular filtration rate for the kidney versus. But my feeling is, I'll be happy to discuss this, is that all tissues age exactly at the same rate because they're aging in exactly the same body. I mean, if you take, for example, the thymus. The thymus is a gland that is very, very active when you're young, involutes when you're young. And so you could argue that that's aging fast, but that's just exactly what the thymus was evolved to do in an animal that lives 100 years. So I think it's actually a little bit misleading to talk about tissues aging faster or slower because something you measure seems to change at a different rate. So, I think it's almost a semantic point, but there are people who would argue that yes, there are tissues that age faster than others, but I think it gets very confusing because there's no gold standard, because you're measuring it against the other tissues in the same individuals aging at a particular rate. So anyway, we can talk later. It gets a little confusing to ask how you define the rate of aging of a tissue as opposed to an organism. Yep.
So maybe in the interest of time, there will be a reception outside, so we can join time for more discussion. So let's thank Tom for a wonderful lecture again.